Okay. So yeah, and I get to tell you about our Crops in Silico project, and then I'm really looking forward to the discussion session so that maybe we can identify some points of collaboration and ways to move the whole field forward. If I go too fast, or if anybody has more questions about the Crops in Silico project, we do have a dedicated website, which is cropsinsilico.org, that you can visit. If you have specific questions for me, you all have my email, but if you want to jot it down real quick, it's right here. Okay. So before I start talking about our virtual crops, I need to talk about the real thing. So we know that 60% of our calories come either directly or indirectly from 5 staple crops, those being corn, wheat, rice, sorghum, and soybean, and I'll be really focusing on soybean today. But most of us are also aware that with climate change, it'll become increasingly difficult to meet the global food demand for these staple crops. So we need new strategies to improve future food security. And this isn't just a casual statement. Just last year, we had some pretty severe weather in the Midwestern United States, and this resulted in unprecedented delays in planting of our major crops, corn and soybean. And then this was followed throughout the growing season with other extreme weather. So for example, there was extreme heat and drought during the critical reproductive phase for corn. And there was also early frost and even snow before the growers could get into the fields for harvest. So all of these things impacted yield of these major crops. And this ultimately resulted in a 5% decrease in yield of corn and soybean nationally, but this was a 20% decrease in yield for the Midwest just from the previous year. And this was the lowest yields that we had had in 5 years. And so this is a really major impact. This translates to millions and even billions of dollars of, to our economy. So when we talk about these strategies to improve food security, our current strategies really focus on trying to understand the relationship between plant genotype and phenotype. And for decades, geneticists have been trying to understand this relationship between genotype and phenotype in order to breed or engineer ideal plants or idiotypes. And so over the years, we've gotten a lot better at breeding for idiotypes because we have just increasing information and access to knowledge that's given us a more mechanistic understanding of plant biological processes. But importantly, we're gaining a clearer understanding of how these biological processes are responding to changes in the environment. And so as our technology increases, we can really take advantage of that. So right now we have great technologies in high throughput phenotyping, gene editing, and high performance computing. And I think that one way that we can take advantage of these technologies to further improve our design of idiotypes is through mathematical modeling. So there are thousands of mathematical models of plants that have been published, and even more studies with empirical data that can be used to parameterize and test these models. And we can think of these models as puzzle pieces that when put together, they make up the full picture of a plant system. So we just need a way to put these together, and that's where the Crops in Silico project comes in. So the central thesis of our project is that we can use integrative and multi-scale modeling to build more accurate representations of crops. And to do this, we have 3 focus areas or components. The first is a computational framework, which is the messaging system or the glue to stick the puzzle pieces together. Then there's the integrated crop models, and we're using these to address specific biological questions. And third, there's also a community component, a community effort where we're looking to engage a broad community of stakeholders that are direct and indirect users, and make this an effort to gain community buy-in and increase communication and collaboration, because no individual lab or even individual project like crops in silico can get everything that we need to build these accurate representations of plants. So it really has to be a community effort. So I'm going to go through and I'm going to talk about each one of these components today, and I'm going to start by briefly talking about the computational framework. And this was developed by Dr. Megan Lang. She's a research scientist at the National Center for Supercomputing Applications. So the motivation to build the crops in silico framework came from the idea that we can combine these existing models to build virtual crops. And the solution that Megan came up with is the Yggdrasil Framework, which coordinates communication between models using APIs and communication files that are either YAML files or JSON files. And the SYS interface has an API, and the models that get plugged into it know how to use that API in order to communicate. What Megan has built in with the SYS interface is that it can also handle data transformation so that users don't have to do it. We feel that this is an easily accessible framework because it's a Python package. And based on community feedback, we've built in certain capabilities and implementation so that we can get a wide user base. So the Yggdrasil framework can be run on Linux, Mac, or Windows. And right now we have support in order to connect models that are written in Python, MATLAB, C, C++, and R. And again, based on feedback that we've gotten from the community, we're currently adding support for Fortran and SBML. And so also, if you're interested in exploring some of the documentation or the tutorials that we have for using this framework, you can visit our GitHub sites that are listed here. And if you don't have time to write these down, I can send that to you. So just shoot me an email. And so another thing that we're trying to do to make this accessible to biologists who dabble in modeling, which is what I would classify myself, is that we're building a user-friendly interface. So in this GUI, we have a drag and drop menu where you can connect models and the input files. And so this is a very sped up version. And this is user-driven. So the files that I was talking about, the connector files, the YAMLs or the JSON, we're also building forms for our users to fill out to generate those files. And so part of that, of the user definition of this, are what are the specific inputs and outputs of their model so that we can drive that communication between the various files. And so that's where we're at right now. So now I'm going to switch gears and talk about how we're using the Yggdrasil framework in order to connect different models and address specific biological questions. And I'm going to be giving you an example in soybean. And this is the work of Dr. Yu Wang, who's a postdoc with me and Steve Long at the University of Illinois, and Kavya Kanan, who was a graduate student with me. She's my first PhD student that just graduated and left, so congratulations to her. And so in this proof of concept that we have, I'll be talking about how we have connected models from the level of gene expression, metabolic flux, and leaf level photosynthesis. And so the proof of concept study is in soybean, and this is because we have a lot of good data at multiple scales in this crop, and that's because it's the second most economically important crop in Illinois next to corn. And its primary use is as animal feed, but it's also an important oil crop for food, biofuels, and industry. And studies at the University of Illinois, we have the Soy Face facility, and I'm sure most of you are familiar with FACE facilities, but they're the free air carbon enrichment experiments. And something that has come out of FACE studies at Illinois and all over the world is that they've shown that many crops, especially C3 crops, have marked physiological responses to elevated CO2. So in this picture, this is the Soy Face facility at Illinois. And you can make out these green rings, which is where the carbon dioxide enrichment is occurring. And this is replicated throughout the field. And the beauty of this is that you have plants right next to your experiment that are under the current ambient levels of CO2. And so this is a wonderful experiment that's been going on for a couple of decades now. And so, like I said, we have a ton of really good information that we can use to put into our models. And so some of the things that have come out from these studies is that leaf photosynthesis in soybean increases 25% across the growing season under elevated CO2 compared to the ambient neighbors. And this was met with a 16% increase in yield in certain genotypes when nutrients and water are not limiting. So this is an interesting result. So under future CO2, predicted or projected CO2 concentrations, we can expect an increase in photosynthesis and an increase in yield. But an interesting thing that has been observed again over the last couple of decades is that US soybean seed protein content has been declining. And this is correlated with the increase or in rising CO2 levels. So something to take into consideration, even though we're getting greater yield, it's not as nutritious as it had been in the past. And then there was another important observation from soy face studies, was that under elevated CO2, there's a decline in rubisco activity relative to the capacity for RUBP regeneration. So even though the light saturated net leaf CO2 uptake, or ASAT, which is on the y-axis in this graph here, while that increases under elevated CO2, which is represented here as the dashed line, so for us, ambient CO2 at the time of this study was 370 parts per million. In the soy face, it was increased to 550 parts per million, which is still the projected CO2 levels for 2050, which is now right around the corner. So we see an increase in carbon assimilation under elevated CO2, but what was surprising here is that it doesn't reach the predicted maxima that of photosynthetic efficiency as predicted by steady state photosynthesis models like the Farquhar model of photosynthesis. So there are some limitations that are being imposed on photosynthetic efficiency even when carbon is abundant. So one thing that has come out of all of these observations from the soy face facility is that we have a hypothesis, is that if there was optimized investment of resources to RUBP regeneration, it could increase ASAT. And so I'm going to get into the details of this in the next few slides from our proof of concept study. But before we could go about even testing this hypothesis, the first thing that we had to do was to build a mechanistic model of soybean photosynthesis that we had faith in before we were able to go in and start doing this scenario testing. So what I'm going to do now is I'm going to take you through each one of the individual models. There are 3 models that we connected in this study. And so I'm going to start off at the most macro scale, which is leaf level photosynthesis. So this model simulates photosynthetic response of soybean leaves to elevated CO2. This can mimic plant response to the field environment, but this is because it's highly empirical. It's not very mechanistic, even though it does have this biochemical photosynthesis component. It's really only the rubisco model of photosynthesis, which only takes into consideration the kinetics of rubisco when there's 170 reactions in the light and dark reactions of photosynthesis. So the major limitation here is that it lacks that mechanistic detail to link physiology and the molecular responses. So it's not going to tell us anything about the mechanism that's driving the observed phenotypes. The other side of the coin is our next model, which is metabolic flux model, and it's an entirely mechanistic model of photosynthesis that represents many of those known reactions in photosynthesis. And this was used in the past, this was developed by Jingwan Zhu. It was used in the past to estimate the optimal distribution of resources in the light and dark reactions of photosynthesis. So, and that's exactly what it can do. It can find those optimal enzyme concentrations theoretically, and the fluxes through the different portions of the pathway. But the problem is here is that it's not at all linked to the physiology, so it can't reproduce some of the phenomenon that were observed in the field for soybean, and especially soybean growing under elevated CO2 conditions. The final component is the gene expression. And so gene expression itself is not the model that I'm talking about, but what we have also from soy face experiments is a transcriptome analysis of soybean growing in ambient and elevated CO2. And independently, that transcriptome analysis suggests that this phenomenon, what I showed you of under elevated CO2, there's an increase in ASAT, but it is still limited. That's called photosynthetic acclimation. And so the transcriptome study suggests that the regulation of that is at the level of transcription. So what my group did is that we took that transcriptome data and we built a new model, and it's a simple translation model that we can take gene expression data and predict protein concentrations based on those relative levels of transcripts that are different between plants that are grown at ambient and elevated CO2. And so that's I guess the benefit of the model is also its limitation, is that it can only estimate relative changes in protein concentrations based on transcript abundance, but it has no link to physiology. So you can see where I'm going with this, is that the independent models describe these individual processes, and they each do a pretty good job at their individual level, but they're all missing parts. There are black boxes in each one of them that limits their usefulness as a really predictive model. So again, the hypothesis is that by integrating these models, we can do a better job of replicating biological phenomenon. And in this schematic, this is showing you some of the information that's passed from one level to the next. So from gene expression, we're estimating protein concentrations that we can then use to update the metabolic flux model. The metabolic flux model produces rates of photosynthesis, including VC max and J max, which can be directly put into the macro level, the leaf level photosynthesis model, which is our link to the environment that feeds information backwards. So here are the results. The major outcome is that our integrated models are, they do a good job of simulating field observed photosynthetic response to elevated CO2. So if we take it stepwise and we just integrate the metabolic flux model with the leaf level model, you can think of what we did is just swapping out the Farquhar model of photosynthesis with the highly mechanistic E-photosynthesis model. And the good thing there is that it didn't ruin the model. So we're still able to get a reasonable ACI curve of carbon assimilation, which you can see here in this graph on the left. But by only including the E-photosynthesis model and the leaf level photosynthesis model, it's blind to exogenous CO2, which is a problem. But when we incorporate information from the gene expression level, now we're able to recapitulate field observations, and now we have this dynamic model that is faithfully responding to elevated CO2. So we're able to predict carbon assimilation under ambient and predicted future levels of atmospheric CO2. So this was goal one, get a model that we can trust. And so that's where we are right now. But then our real goal is to use that model to find ways that we can improve photosynthesis. So now that we have a model that's from the genes to the phenotype, can we use that to figure out how we can modify genes that results in a phenotypic trait that we're looking to achieve? So to do that, we had to go back to the individual models. And the approach that we took for this is that we took that mechanistic model of photosynthesis and used it for what it's good at. So we did a sensitivity analysis of the photosynthesis model to reveal enzymes with high control coefficient. So what are the control points within this pathway that are contributing to those outputs, that VC max and J max? And so after the sensitivity analysis, we identified a number of enzymes. All of our favorites are there, right? Rubisco, FBPase, SBPase, FBP aldolase, and we can break those into the different parts of the Calvin cycle. So we have a couple that are in the carboxylation reactions, those that are involved in the apparatus for RUBP regeneration, and also for starch synthesis. So now we know who those important players are. We know from the literature that the processes that we're interested in are regulated at the level of transcription. So our question then was, now that we know what the control points are, can we identify the transcription factors that is regulating the expression of the genes that encode these enzymes? So we went back to our gene expression data set, and here we did build a different model. This is a gene regulatory network of just the reactions, the photosynthesis reactions, and we used our bioinformatic pipeline to predict transcription factors that we think are regulating them both positively and negatively. And if somebody wants to ask me a question about this, we can during the discussion section. But here in this gene regulatory network, we made it dynamic, where the edges that are connecting these nodes, so I'll just say that the nodes that are in this network, the triangles and the squares, are the genes. The squares represent the metabolic genes that encode our enzymes of interest, and the triangles are all of the transcription factors that are predicted to regulate these genes. That is based on co-expression and also binding motifs and the promoters of our genes of interest. But the edges that connect the nodes, that's where the math comes in. And so here we use training data sets in order to get weights so that we can estimate what is the influence of a particular transcription factor on the expression of a target gene. And so in this model, since it's not static, it's dynamic, we can then tweak the expression of genes in the network, and that signal will propagate throughout the network. So like I did with the photosynthesis model, here we can break this into the different components of the Calvin cycle. So we again have carboxylation, RUBP regeneration, and starch synthesis, where our major control points are. And so now going all the way back to that hypothesis of if we could increase allocation of resources to RUBP regeneration and downregulate Rubisco, we think that we could increase carbon assimilation. So within this network, we were looking for a specific kind of relationship where we're getting repression of reactions over here in carboxylation, but activation of genes, expression of genes that are in RUBP regeneration. And we found several interesting transcription factors, but probably the most interesting is identified as soybean GATA2, because it's predicted to repress expression of the small subunit of Rubisco, the gene that encodes one of those, but activate expression of genes that encode FBP aldolase and starch synthase. So what we did then was we tweaked the expression of that transcription factor. We did an in silico overexpression and an in silico knockout, and then predicted what would be the expression of those target genes. And then we can feed that change in expression into our protein translation model and use that to update the metabolic flux model, which is what we did. And so then once we took this new information that was generated in silico, we put it back into the fully integrated model and then did this scenario testing. And so that's what we're looking at here. This is another ACI curve of if we were to take soybean GATA2 and knock it out or overexpress it. And the red line is the overexpressor, The blue line is the knockout, and the black lines, both dotted and solid, are our wild type. And what we predict here is that by overexpressing GATA2, we can downregulate the part of the curd that represents rubisco carboxylation, and we can up the part of for RUBP regeneration. So we've kind of stacked the deck in our favor, but the phenotypic result of that would be increased carbon uptake rate. So now we have lots of new directions that we can go now that we have this integrated model. Remember that we're only talking about leaf level photosynthesis, and what we really want to get at is whole canopy level photosynthesis so that we can predict changes in carbon assimilation and extrapolate that to yield for a whole field of soybean. And so we're tackling this in a couple of different ways. The first is that we're working with an advanced visualization team to build 3D representations of a soybean canopy. And so this is a data-driven visualization, and it enables predictions about canopy light capture and microclimate with spatial resolution. And so what that means is that we can predict how much light is being captured by every leaf on every plant in an entire canopy of this crop. We can then feed those light capture data into our photosynthesis model and then be able to estimate whole canopy photosynthetic efficiency and carbon assimilation. And then since we have this, this 3D plant that is in this virtual environment, we can then test out different idiotypes, right? We can try different canopy architectures and then do the light capture, feed that into the photosynthesis model. And in that way, we're doing this idiotype testing to see if there's a particular architecture that's going to increase photosynthetic efficiency and again, extrapolate that up to yield. And so next week, you're going to be hearing from a friend and collaborator of mine, James Schnabel. He's a co-PI on the Crops in Silico project. They're tackling this problem in another way that I think will be really interesting to you all. He's working with another co-PI on the grant, Bedrich Bennis, who is a professor at Purdue University, and he uses computer vision. So James has all of these high throughput phenotyping images of corn and sorghum, and Bedrich is using those images to pull out traits that we couldn't traditionally measure. It's not something that we can do with a measuring stick. And so these are all related to voxels and volume and stuff I don't understand. But what's really, really cool about this is that they're able to take these new plant traits that were derived from computer vision and feed them into genome-wide association studies. And I won't spoil it. James will tell you all about the cool results that they found. And then another direction that we're going with this is now we want to swap out that leaf level photosynthesis model for a crop growth model. And so this is work that's being led by Dr. Megan Matthews. She's a postdoc with me and Steve Long at Illinois. And so she's in the final stages of developing a model called Soybean BioCrow, which is a mechanistic crop model that can simulate growth and assess productivity over the course of the growing season. So this is already a multi-component model that uses weather data and empirical field data to parameterize the model. So far, she has optimized the model on data from a single field season of soybean grown under ambient CO2 concentrations, and then was able to predict crop response to elevated CO2 for that same season, and then also for 3 subsequent years. And you can see, looking at these graphs of biomass from the different components of the plant, the leaf, stem, and pod, she's got pretty good estimates, pretty good accuracy, but it's also not a perfect fit. So we think that by including the additional mechanistic details that we can get from gene expression and metabolic flux, we can improve overall prediction accuracy. So those are parts of the next steps for our model integration. We're also working with Jonathan Lynch at Penn State University to integrate our canopy models with his root architecture models. And our goal there is to improve input parameters for nutrient and water uptake, which is what his model called SimRoot does very well. And then just in my last couple of slides, I want to revisit this idea of community science and how we're engaging stakeholders and identifying new collaborations. One way that we do this is that we hold an annual symposium and hackathon, which is an excellent opportunity to interact with people and teach them how to use the framework and immediately get feedback on what we're doing well and what we need to improve on and what new capabilities and implementations we should add. Unfortunately, we're not having it this year. We're postponing it until next year. So hopefully things will be better and you can come to Illinois and visit me. And then the other thing that we've done that I know some of you are familiar with is we've developed a new journal with Oxford Publishing Group that's part of the Annals of Botany family of journals. It's called In Silico Plants. And the goal here is to provide a home for plant models and research related to that, including new tools and software. So if you're interested, I encourage you to look up In Silico Plants. I'm one of the associate editors, so I'd be happy to chat with you about our goals and fit of your project there. And so with that, I want to acknowledge my collaborators on Crops in Silico, my colleagues at the University of Illinois, in particular Steve Long, our partners at the National Center for Supercomputing Applications, which is housed at the University of Illinois, but these are all of these excellent computer scientists that are solving problems that we have no idea how to. And so Megan Lang is very important there. And then we have our collaborators both in the States and abroad. So that's it.